Welcome to Tomorrow Unveiled. Every week, we crack open the cutting edge of science and technology, and, well, right now, nothing seems to be moving faster or carrying more consequence than this whole quest for functional life extension.
It's really accelerating, isn't it?
Absolutely. And we're really talking about health span here. Adding actual life to your years, making sure those years are lived with vitality, mobility, cognition, all of that.
Not just tacking on frail years at the end.
Exactly. Now, this past week, roughly October 8th to the 15th, it might have seemed kind of quiet on the surface. No massive drug launches, no huge headlines grabbing attention.
Right. No single blockbuster announcement.
But our dedicated research team, they've been digging. And the findings they've compiled, well, they reveal this really profound, almost tectonic shift happening just beneath the surface of longevity science. We're seeing these highly corroborated breakthroughs findings popping up across different studies that are genuinely redefining how we understand and maybe even how we could reverse biological aging.
It's less about single discoveries and more about converging patterns, you could say.
That's a great way to put it. So today, our mission is pretty clear. We want to distill those crucial nuggets of insight from this latest stack of research. We're going to unpack three big themes that really stood out.
Okay.
First up, metabolic reset. This is all about those master regulatory pathways that seem to control aging across different species. Really fundamental stuff.
The control knobs, basically.
Yeah. Second, targeted regeneration. This gets into repairing and rebuilding systems we maybe once thought were just irreversibly aged. Think the brain, the vascular system.
Things we assumed were just downhill slopes.
Right. And third, revolutionary technological tools. These aren't just speeding up the research like crazy. They're actually starting to bring personalized longevity into reality.
And that third one really enables the other two in many ways.
Absolutely. It's the accelerator. So maybe you can frame this for us. Looking across all this data our team pulled together, what's the big strategic picture that jumps out?
Well, what's really striking, I think, is this fundamental shift in focus. It feels optimistic, actually. For decades, studying aging was mostly about, decline, observing what breaks down, treating the diseases that come with age.
Pathology focused.
Exactly. But the new focus, the momentum you see in this research, it's shifting rapidly towards actively studying resilience. It's looking at the biology of people who age exceptionally well. Centenarians are the classic example, and asking, what protective mechanisms do they have, and can we learn to mimic them?
So studying health, not just disease.
Precisely. And the underlying principle that this week's findings really hammer home is that aging probably isn't just a random collection of independent breakdowns that we have to patch up one by one.
Like whack-a-mole with diseases?
Kind of, yeah. Instead, the evidence suggests it can be regulated systemically. There seem to be these master switches or pathways. And if we can figure out how to correctly identify and, gently fine-tune those master regulators, well, the whole aging process suddenly looks theoretically malleable.
Okay, that's a powerful idea. Let's unpack that, starting with segment 1. The metabolic reset. If aging is regulated systemically, where are these master control panels? The research our team found points pretty unequivocally towards the conserved TOR pathway, target of rapamycin.
Yeah, TOR is definitely one of the heavy hitters. You could argue it's the most well-understood master regulator of growth and aging we know about. It basically acts like a central metabolic sensor in our cells.
Sensing nutrients.
Exactly. When TOR is highly active, meaning, lots of nutrients and energy are around, it pushes for growth, cell division. But when nutrients are scarce, TOR activity dials down. And that's when the cell switches into this kind of maintenance and repair mode.
Which is why caloric restriction seems to extend lifespan in so many models, right? It keeps TOR suppressed.
That's the prevailing theory, yes. It keeps TOR activity lower. Now, our research team tracked a study on a next-generation TOR inhibitor. It's called Repelink 1.
Okay.
And they found it did prolong the chronological lifespan of yeast, just like the classic drug rapamycin does.
Right, yeast is a common model for this. So if it works like rapamycin, what's new here? What makes Repelink 1 different mechanistically from the 2R inhibitors we already kind of know about?
That's the really interesting bit. Repelink 1 seems to introduce this novel leverage point. The corroborating findings show it works by specifically regulating a set of enzymes. These are called agmatinases.
Agmatinases. Okay, what do they do?
They're responsible for breaking down a metabolite called agmatine. And here's the kicker. Agmatine is produced primarily by your gut microbes.
Whoa. Okay, so hang on. This gets complicated fast. It's not just the drug hitting an enzyme anymore.
Right. You've got this three-way conversation happening. Your gut microbes make agmatine. Agmatinases break it down. And Repelink 1 regulates those agmatinases, which then influences the master aging regulator, TOR.
Wow. That connects the microbiome directly to arguably the most central aging pathway we know.
That's the aha moment, isn't it? It shifts us from just looking at a molecule in isolation to understanding a whole system. The research team's data, it established this direct mechanistic feedback loop.
How so?
Well, when they experimentally caused a loss of agmatinase function in their model system, TOR activity shot up, and that led to premature aging phenotypes. Repelink 1 essentially seems to fine-tune this feedback loop by modulating that gut-derived signal, agmatine.
So the microbiome isn't just like sitting there, it's actively talking to our core aging machinery.
Exactly. It's an active and, importantly, potentially adjustable participant in determining how active this fundamental longevity pathway is. That's deeply significant.
Okay, so stepping back, what does this mean practically?
Mm-hmm.
Say down the line for someone considering a longevity therapy like a rapamycin derivative.
Well, if you connect these dots, it really implies that future TOR inhibitor therapies might not be a simple one-size-fits-all pill. To really maximize how well they work, clinicians might actually need to do a deep dive into the patient's specific gut microbiome. Understand its composition, its function, especially how it handles agmatine.
So you might need a gut test before getting the drug.
Potentially.
Right.
Or it raises the possibility of maybe co-administering specific prebiotics or even engineered microbes alongside the drug. The goal would be to ensure the whole metabolic context is just right for that TO inhibitor to work optimally in that specific person.
That really pushes towards personalized medicine, doesn't it? Moving beyond just the drug itself.
It forces the approach to evolve, yeah, from thinking about a single molecular bullet to aiming for a personalized systemic metabolic tune-up.
That's a powerful validation for a more holistic view. Okay, sticking with metabolism, talk about another major stressor, glycation. This is where sugars kind of irreversibly stick to proteins and lipids, right? Creating AG's advanced glycation end products.
Yeah, essentially gumming up the works. It's a big driver of what you might call diabetic aging or metabolic aging more broadly.
So our research team highlighted some compounds designed specifically to mitigate this glycation stress. They were bundled into something called a glycation-lowering cocktail, or Glylo.
Right, and what makes the methodology here really compelling, I think, is when they gave this cocktail to the mice. It was administered late in life.
Starting around the human equivalent of middle age.
Exactly. Not from birth or youth, but starting later. This timing is critical because it much more closely mimics how a real-world human intervention would likely happen. You don't usually start anti-aging therapies in your teens.
Makes sense. And the results, especially given that late start.
The functional benefits reported were genuinely remarkable. The data showed a 60% extension of remaining lifespan. That's huge.
Wow.
Plus, it preserved lean muscle mass, which is crucial for fighting frailty. And importantly, it normalized glucose homeostasis, hitting that metabolic health aspect directly.
But function is key, right?
Yeah.
You mentioned mobility earlier.
Absolutely. And that's where it got really interesting. They saw a 25–40% improvement in motor coordination. This was meticulously assessed using Rotor Rod tests.
Can you quickly explain what a Rotor Rod test is?
Sure. It basically involves measuring how long an animal, like a mouse, can stay balanced on a slowly accelerating rotating cylinder. It's a really robust measure of neuromotor coordination, strength, balance, things that tend to decline pretty rapidly with age and frailty.
So seeing a 25–40% functional boost late in life is a really strong signal.
It's a huge signal, yeah. And the mechanism they uncovered offers some profound translational potential. It seems the cocktail acts, at least partly, by inhibiting ghrelin signaling in the hypothalamus. Ghrelin is often called the hunger hormone.
Ah, so it's not just acting locally like soaking up sugar damage. It's targeting a master control center in the brain, the hypothalamus, which regulates hunger, satiety, energy balance.
Precisely. By modulating that hypothalamic ghrelin signal, the intervention essentially seems to mimic the positive metabolic effects and the longevity benefits of caloric restriction, which is still kind of the gold standard for extending lifespan in labs.
But without needing the animal, or presumably a person,
Mhm.
to actually cut calories drastically.
Exactly. Without the burden or, frankly, the compliance issues that come with significant overt calorie reduction. So this intervention looks really promising because it hits those metabolic aging phenotypes and delivers tangible functional improvements. Retaining capacity, that's the core mission of health span extension we talked about.
Okay, so if Glylo gives us CR-like benefits via the hypothalamus, that naturally leads us to think about other drugs already out there that impact metabolism at scale,
Mhm.
specifically GLP-1 receptor agonists. Things like semaglutide. This is obviously a massive area right now. Did our sources confirm measurable anti-aging potential here too?
They did. There was a controlled trial involving participants who were on weekly semaglutide treatment, and the finding was that they experienced a reversal of biological age by an average of 3.1 years.
3.1 years.
Based on what?
Based on readings from multiple validated epigenetic clocks. These clocks measure aging at the molecular cellular level. So this finding really corroborates the idea that intervening in metabolism can have systemic reach, right down to our epigenome.
Okay, 3.1 years is a significant number. We know these drugs help with glucose control, weight loss, but why would that show up as such a big reversal on an epigenetic clock? What's the connection there?
Well, what's particularly fascinating is where those anti-aging benefits seem to be most concentrated. The reversal wasn't necessarily uniform across all tissues measured by the clocks.
Oh. Where was it strongest?
It was most pronounced in two key systemic areas, the inflammatory system and the brain. In those specific biological systems, the clock suggested biological aging was delayed by almost 5 years.
Wow, 5 years in the brain and immune system. Yeah, and this ties back perfectly to that idea of the systemic nature of aging. GLP-1 agonists are known to significantly reduce chronic inflammation, that inflammaging concept that seems to drive so much age-related pathology.
Right.
And they lower the overall metabolic burden on the body. Together, this seems to ease the strain on all the body's aging systems, but maybe particularly on the highly sensitive neural environment and the constantly active immune system. It really confirms that easing metabolic stress has these profound, widespread anti-aging effects that we can now actually start to see and measure with our most sophisticated molecular tools.
That really sets the stage perfectly for our next segment, segment 2. Regeneration and restoration. We're moving from metabolic control, dialing things back, to actually repairing the physical infrastructure. Let's maybe start with the supply lines, the vascular system and how its decline contributes to physical frailty.
Yeah, that's a great place to start. We often focus directly on the muscle itself when we talk about sarcopenia, age-related muscle loss. But the research team highlighted findings emphasizing the critical importance of the infrastructure supplying that muscle, specifically the endothelial glycocalyx.
The glycocalyx. That sounds technical. What is it exactly?
Think of it like a very delicate, fuzzy, almost gel-like lining on the inner surface of every single blood vessel, from the biggest arteries down to the tiniest capillaries.
Okay, like a coating.
Kind of, but it's incredibly important functionally. It acts as a primary filter, regulating what substances get absorbed from the blood into the surrounding tissues. It also plays a huge role in controlling local inflammation and blood flow.
And this lining degrades with age.
Yes, quite rapidly, unfortunately, and its degradation is now being directly implicated as a contributor to frailty and sarcopenia.
So the idea is, if the glycocalyx breaks down, the muscle factory is essentially being starved of nutrients and oxygen, and may be bathed in inflammatory signals, even if the main highway, the artery itself, isn't completely blocked.
That's a perfect analogy. The muscle is the factory, the blood vessels are the highways. The glycocalyx isn't the road itself, but it's like the crucial on-ramp and maybe the customs agent controlling what gets delivered.
Got it.
When that breaks down, delivery is impaired, waste removal slows down, and localized inflammation can spiral. So, the intervention highlighted here involved giving aged mice a dietary supplement. It was rich in something called high molecular weight hyaluronan, HMWHA, for about 10 weeks. The goal was to try and physically restore that degraded glycocalyx layer.
HMW-HA,
Yeah.
like hyaluronic acid used in skincare.
Related, yes, but specifically the high molecular weight form, which seems to have different biological properties relevant to the glycocalyx structure.
And did it work in the mouse model? Were the functional benefits robust?
They were, yeah.
Mm-hmm.
According to the data, the treated mice preserved more muscle mass compared to controls. They maintained a higher maximal exercise capacity. And they showed better markers of mitochondrial health, like citrate synthase activity, right there in the muscle tissue.
So the idea holds up. Improve the vascular supply lines, and the muscle factory operates more efficiently, keeping its energy production up and preventing atrophy.
That's the concept, and the mouse data strongly supports it.
Okay, now this intervention, as noted in the sources, uses a supplement pathway. There's a specific product mentioned, Endothelial. Supplements can get to market much faster than drugs. But you hinted at caution earlier. If the mouse data is that compelling, why should a consumer maybe pause before jumping in? Isn't the evidence clear?
Ah, that's such a crucial point, and it really exemplifies what we call translational friction, or the translational gap. While that mouse data is incredibly robust, showing really dramatic functional gains, human pilot studies using these HMWHA supplements have, so far, shown pretty mixed results, often equivocal.
Why the difference?
Well, the challenge is complex. For the supplement to work, the HMWHA has to survive your digestive system and needs to be absorbed effectively into the bloodstream, and then it has to actually reach the endothelial cells lining the blood vessels intact in a high enough concentration to rebuild the glycocalyx.
And that might not happen efficiently in humans.
It seems less reliable. The human gut environment, our metabolism, it might neutralize or break down the compound much more effectively than in a lab mouse. This really highlights that significant gap. What works brilliantly in a highly controlled, genetically uniform mouse model doesn't always translate reliably into a complex, genetically diverse human population with varied diets and lifestyles.
So consumers need to be aware that the marketing claims might be heavily based on that preclinical promise.
Exactly. Based on the promise, not necessarily on fully validated human efficacy at the same level, it's a real challenge in navigating the supplement space for longevity.
That's vital context to keep in mind.
Yeah.
Okay, let's shift from supply lines to actually cleaning up the cellular environment. This involves one of the hottest areas in longevity right now, Senolytics. Targeting those toxic, non-dividing zombie cells. Our research team detailed a pretty bold intersection of this senescence targeting idea with acute medicine, specifically in neurological repair.
Yes, this was fascinating. A longevity biotech company proposed a novel kind of two-step approach. They're exploring it for potentially restoring consciousness in patients with severe brain injuries or even those in a coma.
A two-step approach. How would that work?
The idea is, first, you'd administer a senolytic therapy, likely an immunotherapy designed to target and clear out senescent cells specifically in the injured brain area. Then you'd follow that up with an infusion of the patient's own mesenchymal stem cells.
Okay, so the underlying implication there is pretty huge. It suggests that severe brain injury, or maybe even advanced neurodegenerative aging, doesn't just involve dead or dying cells. It actually creates a toxic environment, right? Full of these inflammatory signals from senescent cells, the SAP or senescence-associated secretory phenotype.
Exactly. And that toxic environment actively blocks natural repair processes and maybe hinders the effectiveness of things like stem cell therapy. So the thinking is you have to clear out the junk, neutralize the toxic environment first before you bring in the repair crew, the stem cells, to give them a chance to work effectively.
That concept seems like it could apply more broadly, too.
It does seem to hold up elsewhere. Separately, there was data from a phase 2 trial in cancer patients. They combined senolytics with standard immunotherapy. And they found it not only reduced the toxicity of the cancer treatment, but also achieved a measure of functional rejuvenation.
Rejuvenation in what sense?
Critically, for our discussion on aging, it reversed markers of immune aging. Specifically, it helped restore the function of crucial naive T cells, which decline significantly with age and are vital for responding to new threats, like infections or even cancer cells.
So cleaning up senescent cells can actually improve the quality and youthful function of the immune system systemically.
That's what this data suggests, yes. It confirms that cellular cleanup isn't just about removing bad cells. It can genuinely rejuvenate systemic function, improving the quality of vital systems like immunity.
Okay, speaking of systemic rejuvenation, let's talk about therapeutic plasma exchange, or TPE. This has gotten a lot of buzz, maybe some hype too. What did the actual corroborated data that our team looked at show regarding reversing biological age?
The finding was pretty unambiguous, actually. TPE alone, just the plasma exchange process, was shown to significantly lower biological age in adults over 50.
And how much lower?
When they assessed it using a panel of 36 different validated epigenetic clocks, TPE on its own reduced biological age by an average of 1.32 years.
Okay, over a year, that's noticeable,
Yep.
but there was more to it.
Yes. When TPE was combined with IVG, that's intravenous immunoglobulin, the reduction in biological age jumped significantly to an average of 2.61 years.
Wait, let's pause there. TPE, we know, involves physically removing a person's blood plasma and replacing it with saline and albumin.
Yep.
The idea is to dilute and remove inflammatory factors.
Exactly. You're washing out pro-aging factors, circulating inflammatory proteins, things like that.
But what is IVG? And why would adding that make such a big difference? That's almost double the effect.
Right. That synergy is where the molecular insight lies. IVE is essentially a purified blood product derived from thousands of healthy plasma donors. It consists mainly of pooled human antibodies, IgG antibodies specifically.
Okay.
It's often used clinically to treat immune deficiencies because it provides this broad, natural immune modulation. It's like giving the body a massive dose of diverse regulatory antibodies. So the thinking is, TPE removes the bad stuff, the inflammatory, age-associated proteins driving inflammaging. Then, IVG effectively replaces the plasma not just with saline, but with immunomodulatory good stuff.
So you're clearing out pro-aging signals and actively boosting the regulatory capacity of the immune system at the same time.
Precisely. By doing both, the combined approach seems to achieve a deeper, more profound biological age reversal than TPE alone.
Interesting. And did everyone benefit equally, or did it work better for some people?
The research indicated that individuals who started with poorer baseline health markers, things like higher circulating glucose levels or higher baseline inflammatory markers, they actually experienced the greatest rejuvenation effect from the TPE-IVIG combo.
So it's particularly effective at reversing accumulated measurable damage.
Yeah.
Maybe more so than just tweaking an already healthy system.
That's what the data suggests, yes. It seems most impactful for those who have more aging damage to clear out, so to speak.
This evidence of systemic repair, washing the system clean, it leads us nicely into thinking about really precision repair in specific systems, like the nervous system. True targeted regeneration. Let's start with vision. Our research team noted a finding about PUFAs, polyunsaturated fatty acids, and age-related vision decline.
Yes, this was a really elegant piece of research, I thought. They found that supplementing aged mice with one specific PUFA actually reversed age-related vision decline.
Reversed it? How?
The mechanism is quite fascinating. It seems to work by essentially bypassing the function of a gene called ELOVL2.
EOVLO2. Okay, what's its significance?
ELOVL2 is known to be an aging gene. Its activity naturally declines quite significantly with age. And this decline leads to a shortage of certain downstream fatty acid products that are absolutely critical for maintaining the health and function of retinal cells in the eye.
Ah, so instead of trying risky gene therapy to somehow boost the declining ELVL2 gene back up,
Right.
they just supplied the missing product that the older system couldn't make efficiently anymore.
Exactly. It's a very pragmatic restoration strategy. You identify the bottleneck caused by aging, and you provide what's missing downstream. And the functional benefit reported was clear. Improved visual performance in the aged mice, and they could even see a reversal of some molecular features of aging right there in the retinal cells.
That's a really targeted restoration mechanism, addressing the consequence of an aging pathway without needing to directly fiddle with the gene itself.
Precisely. It's quite clever.
Okay, and what about the brain itself? We often hear about protecting neurons, but what about actually making new ones? Neurogenesis, was there anything exciting there?
Yes, definitely. Researchers reported synthesizing some new supercharged vitamin K analogs.
Supercharged vitamin K, how?
They essentially designed molecules that combine structural features of both vitamin K and retinoic acid. And they found these new hybrid analogs were about 3 times more potent than natural vitamin K at inducing neural progenitor cells. Those are like the brain's dormant stem cells to differentiate into new, mature neurons.
Okay, stimulating neurogenesis more effectively, that's interesting. But the big hurdle is usually getting things into the brain, right? The blood-brain barrier.
Absolutely critical point. And that's what made this finding particularly exciting. These new analogs were shown to efficiently cross the blood-brain barrier in their mouse models.
That is huge, because the blood-brain barrier is like the ultimate gatekeeper, right? It stops most things we take systemically from ever reaching the brain tissue in significant amounts.
It does. So finding something that not only gets across that barrier efficiently, but also acts as a potent regenerative trigger, stimulating the birth of new neurons, not just protecting the ones you already have, that's really foundational. It offers the potential capacity to actually rebuild lost neural connections.
This really shifts the strategy, doesn't it?
Yeah.
from just neuroprotection, trying to slow the decline, towards genuine neuroregeneration, maybe opening the door to reversing damage in conditions where neuron loss is the key problem, not just slowing things down.
That's the hope, yes. It's a fundamentally different, more proactive approach.
Okay, this seems like a good moment to remind everyone listening. If you're finding this insightful, please take a moment to like this video, subscribe to Tomorrow Unveiled, and maybe consider hitting that new hype button on YouTube. Apparently, it helps us reach more viewers. And of course, do turn on notifications so you never miss an update on the future.
And for our audio listeners.
Right. If you're listening on a podcast service like Apple Podcasts or Spotify, we'd be incredibly grateful if you could leave us a rating and maybe a quick review for Tomorrow Unveiled. It genuinely helps new listeners discover the conversation.
It really does make a difference.
Okay. Welcome back. We've covered the metabolic reset. We've talked about regeneration and restoration. Now let's dive into segment 3. Technological enablers and regulatory shifts. These are the tools and the rules shaping how quickly this field moves. And let's start with what might be, strategically, the most significant development for future drug discovery: the Centenarian IPSC platform.
Yeah, I agree. This is potentially transformative. It's an enabling technology that could seriously accelerate the entire field of discovering drugs for human longevity. Our research team confirmed the establishment of this platform using induced pluripotent stem cells, IPSCs, derived directly from centenarians.
People over 100 who've stayed remarkably healthy.
Exactly. Those exemplars of exceptional health span.
So why is using their stem cells so critical? I mean, we've been studying centenarian genetics for years through things like GOBS, right? Genome-wide association studies. What does making IPSCs let us do that just sequencing their DNA couldn't?
That's the key difference. GWS studies are great at finding statistical associations between certain genes and longevity. But there's always been this huge bottleneck in linking those genetic associations to a specific causal effect within a living cell.
How does the gene actually do something protective?
Right. The iPSC platform closes that gap beautifully. You can take a blood cell or a skin cell from a centenarian, use specific factors to reprogram it, push it back to this primitive, embryonic-like stem cell state, the iPSC state.
Yeah.
And then the magic is you can differentiate that iPSC into basically any specialized cell type you want to study: neurons, heart muscle cells, liver cells, immune cells, you name it. And crucially, all those derived cells will carry the unique genetic and epigenetic signatures of that individual's exceptional longevity.
So you get a living, functioning human cell model of healthy aging in a dish.
Exactly. A scalable, experimentally tractable human model. Not a mouse model, not just correlations from genetics, but actual human cells exhibiting resilience.
And what are these centenarian-derived cells telling us so far? Are we actually seeing some kind of common resilience signature emerging?
We are. The early results highlighted by our team show that neurons derived from these centenarian iPSCs exhibit really distinct functional signatures compared to neurons derived from, say, typical elderly controls.
Like what?
Specifically, they seem to show enhanced synaptic integrity, meaning the connections, the synapses between the neurons are more robust, better maintained. And they also show a notably more efficient and robust response when faced with cellular stress, like oxidative damage or protein misfolding. They handle stress better.
So this platform is basically set up for drug screening now.
Absolutely. It's primed for high-throughput screening. You can expose these resilient centenarian cells and maybe control cells to thousands of different molecules and see which compounds can make the control cells behave more like the centenarian cells. The ultimate goal is to find drugs that can pharmacologically induce this innate cellular resilience signature. Essentially give everyone's cells a bit of that centenarian-like defense mechanism.
The pace of discovering those molecules, though, it depends on powerful tools. And that brings us neatly to the rise of AI, digital twins, and personalized health span. We're talking about tech that's moving way beyond just analyzing data after the fact, right? It's getting into real-time personalized intervention.
It really is. Our research team noted multiple instances where AI platforms are just dramatically compressing research timelines. For example, there are reports of using multi-agent AI systems to analyze these vast, incredibly complex, disparate data sets: genomics, proteomics, metabolomics, lifestyle data from wearables.
All crunched together.
Right. And these AI systems can apparently build novel biological aging clocks, identify new biomarkers, or even propose intervention strategies in a matter of weeks. Tasks that would honestly take traditional human research teams months, maybe even years, to coordinate and compute.
And these sophisticated AI tools, they're not just staying in the deep research labs anymore, are they? They're moving into actual daily health management through things like digital twins.
Exactly.
So,
We're seeing AI models, often in collaboration with health tech platforms. The sources mention Twin Health as an example that are integrating real-time dynamic data streams. Think continuous glucose monitors, CGMs, sophisticated activity trackers, sleep monitors.
all feeding into a model of me.
Precisely. Feeding into a model to create what they call a virtual metabolic replica, or a digital twin. It's a dynamic simulation of your specific metabolic environment, constantly updated with your real-world data.
So the practical application here isn't just getting a fancy report card at the end of the month. It's about real-time course correction.
That's the goal, yes. These twins enable potentially ultra-precise, often non-drug interventions that are tailored specifically to your unique biology in that moment.
Give me an example.
Okay, so maybe your digital twin analyzes your sleep data from last night, your current glucose trend from your CGM, and your planned workout. It might predict that, based on your poor sleep, your usual morning brisk walk is likely to cause an inefficient metabolic response, maybe a big glycemic spike.
Okay.
So the system might advise you to tailor that exercise session, maybe switch to lower impact cardio today, or perhaps focus on resistance training instead, specifically to mitigate that predicted spike that the twin foresaw based on your personal data.
So it's optimizing diet, exercise, sleep, stress management
Right.
with this extreme, granular, real-time precision based on your own body's signals.
That's the vision, using AI to achieve a level of personalized optimization that's just not possible otherwise.
Okay, fascinating stuff on the tech front. Let's move from the enabling tech to the rules of the road. Regulatory precedents and challenges. This is crucial for getting these breakthroughs to people safely. I want to highlight something our team flagged as the mitochondrial milestone of the week. It might seem a bit niche initially, but the strategic implications for the whole longevity field could be enormous.
I agree, this is highly significant. The FDA granted accelerated approval to a drug called Forzinity. The generic name is lamipretide. It's a drug specifically designed to target mitochondrial dysfunction.
Okay, mitochondria, the powerhouses of the cell.
Yeah.
But the approval was for a rare genetic disease, right? Something called Barth syndrome.
That's correct.
So why does getting approval for a rare disease matter so much to the broader field of longevity, which is focused more on generalized age-related decline?
Because this marks the first formal regulatory acknowledgement by the FDA that targeting fundamental mitochondrial health can actually translate into measurable, clinically meaningful human therapeutic benefits.
What benefit do they measure?
In the clinical trial for Barth syndrome, the drug showed an improvement in leg muscle strength and function. And the FDA accepted that functional improvement, things like strength, mobility, exercise capacity, as a valid clinical endpoint for a drug targeting mitochondrial biology.
Ah, so it validates the scientific rationale.
Exactly. It validates the rationale behind literally dozens, maybe hundreds of other anti-aging efforts currently underway that are aimed at mitigating mitochondrial decline. Because mitochondrial dysfunction is implicated in almost all major age-related conditions: frailty, sarcopenia, heart disease, neurodegeneration, you name it.
So this sets a really important regulatory precedent. It says, yes, improving mitochondrial function is a valid therapeutic target, and yes, improved physical function is a valid outcome to measure.
Precisely. It paves the way for future submissions targeting broader age-related mitochondrial decline.
Okay, that's a positive sign on the regulatory front. But on the flip side, we also saw a critical development that highlights the challenges.
Yeah.
The NMN Reversal.
Yes, the critical contrast. The FDA essentially reversed its previous position on nicotinamide mononucleotide, or NMN. They formally stated that NMN is now excluded from the definition of a dietary supplement under the relevant act.
Excluded. Why?
Because NMN is currently being investigated as a new pharmaceutical drug. The FDA's reasoning is that if a substance was not marketed as a supplement before substantial clinical investigations as a drug were instituted and made public, it can't later be classified as a supplement. And more fundamentally, the implication is that if something acts like a drug,
Meaning it targets specific biological pathways to achieve a therapeutic or preventative health benefit.
right, if it acts like a drug, it needs to be regulated like a drug. That means going through the full, rigorous process of clinical trials to prove safety and efficacy for specific indications, getting FDA approval, etc.
Even though NMN is still widely available to buy online right now,
Yes, the enforcement is a separate issue, but the FDA's official stance sets a clear line. It underscores the high standard, the absolute need for validated human functional data, not just animal studies or mechanistic theories that these translational findings must meet to deliver reliable, safe health span benefits. It's a pushback against the market being potentially flooded with compounds marketed for anti-aging without that rigorous proof.
This whole blurred boundary between supplements and drugs, it actually leads us directly into our final segment, segment 4, ethical and practical considerations. We have to balance the genuine excitement around these breakthroughs with the tricky realities of implementation, especially concerning consumer risk and equitable access.
Yeah, this is a really necessary part of the conversation. The first major point of friction revolves around exactly what you just mentioned, the regulatory gray zone and consumer risk.
The examples we talked about, like the Glycocalyx targeted therapy, HMW-A, or even NAD plus precursors like NMN or NR, they really exemplify this dilemma, don't they? Our research sources consistently show incredibly robust, sometimes really exciting data in animal models.
Often dramatic effects in mice or worms or yeast.
Right. And that animal data then gets used quite extensively sometimes in marketing materials, promising profound benefits to humans. Yet when you dig into the human clinical data for these same compounds used as supplements, it often remains equivocal or shows much more limited effects, maybe highly variable effects between individuals compared to what was seen in the tightly controlled animal studies.
It puts the consumer in a really challenging and sometimes quite expensive position. You're essentially being asked to purchase products based largely on preclinical promise, maybe some mechanistic rationale, rather than on solid proof of human efficacy and optimized dosing for longevity outcomes.
And the supplement pathway, being faster and less regulated, kind of enables this.
It does. While that fast track can seem exciting for getting innovations out quickly, it often sacrifices the rigorous multi-phase validation that's required to ensure that the functional health span benefits seen in the lab animals actually translate reliably and safely to humans. Plus, there are often issues with quality control, dosage consistency, and purity in the supplement market that you don't typically see with approved pharmaceuticals. It creates a real financial risk and potentially a health risk, too, for consumers eager for genuine longevity solutions.
We also absolutely have to consider safety and off-target effects. When you start talking about modulating really fundamental, highly conserved biological pathways, things like the TOR pathway we discussed, or deliberately clearing out entire populations of cells like senescent cells, the potential for unintended systemic consequences for off-target risks is inherently high, isn't it? It feels like a double-edged sword.
It absolutely is. Take TOR inhibition, for example. We know that dialing down TOR can promote longevity in many models, but TOR also plays critical roles in things like immune function, wound healing, and muscle protein synthesis.
So over-inhibiting it could be bad.
Potentially, yes. If you suppress TOR too much or at the wrong time, you could theoretically impair your immune response to infections, slow down recovery from injury, or even interfere with maintaining muscle mass, especially under certain conditions like illness or high physical stress. For a relatively healthy person seeking prevention, those potential off-target effects are a major concern that needs careful study.
And the complexity we saw with agmatine and the microbiome feeding into TOR.
Exactly. That discovery that the metabolite agmatine plays this complex regulatory role, it strongly cautions against the sort of indiscriminate use of any purported anti-aging supplement that might interact with this core pathway, especially if you don't understand the individual's specific metabolic context, like their gut microbiome's activity.
We need deep safety data.
We absolutely need rigorous confirmation of safety, especially long-term toxicity profiles, before these kinds of fundamental interventions are applied broadly to relatively healthy populations just for prevention or general anti-aging. Are we inadvertently solving one age-related problem in the short term only to potentially create new vulnerabilities, maybe in the immune system or tissue repair, decades down the line? That has to be thoroughly investigated.
That focus on long-term safety for preventative use is critical. Okay, finally, let's touch on the huge issues of accessibility, equity, and privacy. If these powerful new longevity breakthroughs, TPE, stem cells, advanced AI coaching, only end up benefiting a small, wealthy fraction of the population, we risk creating a really severe two-tiered health system, don't we? Where functional longevity becomes a luxury commodity.
The cost barriers are immediate and they are significant. Take therapeutic plasma exchange. As we discussed, it shows promise for biological age reversal, but it requires specialized apheresis equipment, trained clinical staff, sterile facilities.
And not a simple pill.
Not at all. Each session can cost thousands of dollars, often exceeding $5,000 per treatment was mentioned in some contexts. And currently, it's generally not covered by insurance for longevity or preventative purposes, only for specific approved medical conditions.
And that's likely just the tip of the iceberg.
Probably. Emerging stem cell therapies, any future gene editing techniques that might arise, even the sophisticated AI-driven digital twin services we talked about, they will almost certainly start out as being prohibitively expensive for the vast majority of people. So, we have to ask the hard question now. How can these potentially life-altering functional longevity tools be democratized? How do we prevent them from just exacerbating existing health and socioeconomic inequalities?
And the very technologies we celebrated earlier, the IPSCs and the AI twins, they also raise some pretty significant ethical flags around privacy and control, don't they?
They certainly do. That Centenarian IPSC platform, while scientifically incredible, it brings up complex questions. Things like ensuring truly informed consent from potentially frail, very elderly donors. And then who owns the genetic secrets of longevity that might be discovered from their cells? How is that information shared and used?
Data ownership is a big one.
Huge. And similarly, the AI digital twins rely on integrating absolutely vast amounts of highly personal, highly sensitive biometric and metabolic data, often collected continuously in real time.
Your entire biological life streamed to the cloud.
Pretty much. So we absolutely must ensure there are robust, maybe GDPR compliant or even stronger frameworks in place. Things like genuine, unambiguous opt-in consent, strong data encryption, and secure anonymization options are critical to protect individual privacy, especially as these systems potentially transition from being just helpful wellness tools to becoming essential guides for managing personalized health and longevity strategies. Trust is paramount.
Absolutely critical points to consider. Okay, as we start to wrap up this edition of Tomorrow Unveiled, let's try to recap the central finding from this week's research stack. For me, it feels like the science isn't just advancing incrementally anymore, it's genuinely maturing.
I think that's the undeniable truth, yes. We saw clear, corroborated breakthroughs across multiple fronts. In metabolic regulation, the really specific insights into how the microbiome fine-tunes TOR via mechanisms like Repelink 1, and the tangible functional benefits seen with the Glylo compounds mimicking caloric restriction effects. Then in restoration, we saw significant advancements, from the potential of supporting vascular health via the Glycocalyx, even with the translational caveats, to the quite striking systemic rejuvenation suggested by the TPEI-VIG synergy in reversing epigenetic age.
And underpinned by the tech.
And all of this is clearly being accelerated by these powerful enabling technologies, most notably that Centenarian IPSC platform providing unprecedented human models, and the AI-driven digital twins starting to offer real personalized insights. The convergence is palpable.
And it feels like the scientific focus itself is undergoing that shift we talked about at the start, moving away from just studying generalized pathology and towards understanding specific mechanisms of resilience.
Exactly. I think the field is successfully making that pivot, studying the actual biology of exceptional health and not just the biology of decline. The next critical steps, though, will be all about translation, determining how these incredibly promising strategies that we see working in models, whether it's vitamin K analogs boosting neurogenesis, or Repelink 1's precise mechanism, or Glycocalyx support how they get successfully validated in rigorous human trials.
With a relentless focus on function.
Absolutely. Focusing relentlessly on measuring meaningful functional outcomes. Mobility, cognitive function, independence, quality of life. Because ultimately, the future of longevity has to be function first, adding healthy, capable years.
And that focus on function, on extending health span potentially by years, maybe even decades eventually, it brings us to a final, really profound thought for everyone listening. If we do succeed in compressing morbidity, in keeping people healthier for longer, we absolutely must ask ourselves, are we as a society actually prepared for the profound implications?
Yeah, the downstream effects are huge.
Think about it, the impact on retirement structures, on workforce dynamics, if people can work productively for much longer, on intergenerational relationships, even on the very definition of what constitutes a meaningful life if a healthy 100-year lifespan becomes common. Are we ready for a large, healthy population of centenarians? That's the big question we leave you with mull over this week.
A lot to think about there.
Definitely. And on that note, we really do love hearing your thoughts. Whether you're watching on YouTube or listening on your favorite podcast app, please leave a comment. Let us know what you think about this Immortality update. What excites you? What concerns you? Especially around those ethical questions of access, safety, and that regulatory gray zone we discussed.
We do read the comments.
We do. Thank you so much for joining us for Tomorrow Unveiled.